To be honest, I haven't thought about neurons and all of this brain kind of stuff since the time in high school where I tried to make sense out of what my biology teacher was desperately trying to explain to me. And even though artificial intelligence has become quite a buzzword in the past few years or maybe even decades, my understanding of what you can do with it really stops at you can build a cute humanoid robot with it. Thanks for that, Steven Spielberg. But both of those topics are very important in our society and our whole entire world. So knowing a bit more about it really wouldn't hurt. And with this, welcome everybody to the 5th episode of the Science Slam Challenge. My name is Emma Miesler and I will be your host for today, guiding you through today's guest's presentation on exactly those two topics, AI and neurons, and what wonderful things you can do with the combination of those two. But before we go more in depth about today's topic, and before I reveal today's guest to you, let's maybe explain the concept of the show to people who have never watched an episode of the Science Slam Challenge. But since me talking all the time would be maybe a bit tedious, we have prepared a short explanation video for you.
In every episode of the Science Slam Challenge, a researcher presents a self-chosen topic they are currently researching on. Be it medicine, English studies, or history, the research field really doesn't matter. Today's guest has, as always, 3 times 3 minutes to explain his or her research to us. The challenge is to make those presentations as compact, comprehensible, and entertaining as possible. Our moderation will ask some further questions in between those presentation parts. Got it? Then, without further ado, let's start with today's Science Slam Challenge.
Sounds easy, right? So now I can finally reveal today's guest to you, the neuroscientist Yves Bernaerts. Yves, welcome. I'm so happy you're here.
Glad to be here.
Yves, in our initial meeting, you told me you and your team are trying to figure out how neurons work. But if I remember correctly from school, they send out electrical signals and that's how they communicate, that's how they work. Is there really so much more to research on?
Yes, so there's indeed different angles with which we can view neurons. There's indeed those electrical signals that you already correctly pointed out, but there's also gene expressions and how they vary amongst neurons, and they also might look, some neurons might look very similarly, other neurons might actually look very dissimilar to those neurons. So there's different angles to view neurons with, yeah.
I'm very excited to get to know more about that, and I see you're equally as excited to talk. But I think it would be very interesting for our viewers to get to know you a bit more better. And for that, we have prepared a short video.
Yves Bernaerts works as a neuroscientist at the University of Tübingen. He first came from Belgium to Tübingen during his master's as an Erasmus student. He enjoyed it so much that he stayed here to work on his PhD in neuroscience. With a bachelor in physics and a master's in biochemistry, physics and technology, the combination of his two fields of interest seemed to be the next logical step. In Tübingen, he and his team are mostly focused on artificial intelligence, the human brain and neuroscience in general. More precisely, the question how artificial intelligence might benefit us on trying to comprehend the human brain and broaden our understanding of the way neurons work are of a particular interest to them. Today, he is giving us a glance into his work and takes us on a trip through the human brain. A warm welcome to Yves Bernaerts.
So now that everybody got to know you a bit more better, I think we are ready and set to start today's Science Slam Challenge. Yves, if you're ready, your time starts now.
All right, thank you. So my job on one level today would indeed to try to give an overview of the research that we're working in at this intersection of machine learning and neuroscience, but on another level also to uncover this box with the buzzword machine learning on it and give an actual flavor of what we're doing in machine learning in this particular setting. So it's called Making Sense of the Human Brain with AI. And indeed, as humans, aren't we interested in our own human brain? In fact, we're so interested in it that the Obama administration in 2013 launched the Brain Initiative. So a very adept name. It's indeed an initiative to revolutionize our understanding of the brain, and with that perhaps also treat patients with, for instance, Alzheimer's or Parkinson's better. Within this big initiative, there's a big project that is called the BICCN. It stands for the Brain Initiative Cell Census Network. And so it seems to be about cells. And indeed, this big project starts from the perspective that, well, if we're trying to understand our brain, maybe we should try to understand its constituents, its building blocks first. And those are indeed cells, specialized cells, neurons. And those neurons, it turns out, can be approached from different angles or viewpoints. There is, for instance, the genes. There might be a certain set of neurons that expresses a certain set of genes very highly, while there might be another set of neurons that actually oppresses those same sets of genes. So that's already a variety in how genes can be expressed in different neurons. That's one angle, one viewpoint. Another viewpoint might be the electrical behavior. So maybe these electrical dots that you could see here, those are electrical signals which propagate from one neuron to another. And in fact, an electrical signal is nothing more than a voltage that exists on the outer layer or membrane of the neuron, and which can then fluctuate also in function of time, depending on the input that this neuron receives. And it turns out that, well, certain neurons, they behave similarly electrically, but other neurons might actually behave dissimilarly from them. And then there's also their morphology. Again, certain neurons might look very similarly in its morphology, but another set of neurons might look very dissimilar to them. So you have certain types, but then you also have perhaps a continuum of types. So this is really research that we focus on, that we're researching. It's these different viewpoints of same neurons.
Wow, that was actually perfect on the second. Thank you so much for your first explanation, Yves. I actually do have some questions about that. In the beginning, you were talking about this like big collaborative effort. What exactly is your research group's like role in this project?
Yeah, so those different viewpoints that I talked about. So we have collaborators in Houston, in Texas, and they've done a tremendous job of collecting the data, those gene expression levels that I talked about, but also the electrical behavior, and even also the morphology. And we, as data science in Tübingen, we got the job to analyze that data and also to actually try to relate different viewpoints from, say, for instance, genes, and try to relate that to the electrical behavior.
Coming to the electrical behavior, you showed like a graph. There were like some peaks, and at some point it was more like stable. So why does that happen? What does that exactly mean?
Yeah, good question. So our experimenter in Houston, the experimenter can take its pipette and go to the neuron, get really close, and then decide to, for instance, inject some negative charges. That would be a negative current. And how then this voltage would respond is seen here by this blue curve. So it would go down, and then it would receive, or it would reach a steady state, we call it again, until the experiment is done, and the membrane voltage stabilizes again at its natural level. If it would decide, if the experimenter would decide to inject some small positive charges, then it goes up a little bit, reaches a steady state for during the whole experiment again, and go down. If, however, the experimenter introduces a lot of positive charges, then the neuron will actually arrive at its threshold and start showing this peaking behavior. I hope that answers your question to a certain extent.
It actually does. And I think now we're ready to start with the second part of your presentation. Yves, if you're ready, we're ready. The timer starts now.
All right. So let us now maybe focus on two viewpoints, two angles, the genes and the electrical behavior. So now one might ask, how can now machine learning or artificial intelligence help us to understand neurons? Well, it could perhaps figure out a mapping, a function, f, if you'd like, that takes in gene expression levels and outputs the characteristics that describe this electrical behavior. So input genes, output electrical behavior. Can we do such a thing with machine learning? Let us maybe first look at what we call a toy example, a simpler example. Say we have two variables, x and y, and x might predict y, and we've collected data points. They can be seen here in blue, and maybe we think that a linear fit, the red line here, would be good to explain the data. However, if we look at it, maybe you would agree with me that we're not doing such a great job yet. In fact, we could somewhat calculate that. If we calculate the distance from this blue point towards the red line, well, we can do that also for this point. And for this point, we can in fact do that for all the points. We could sum up those distances and we would arrive at a certain value. It could be interpreted as an error value, in fact. And that is exactly what a machine learning algorithm could do. It could try to reduce that error to a very minimal value. Here you can see it slowly, step by step. Our machine learning algorithm finds a better and better fit. In fact, at iteration 15, you might agree with me, it starts again, but that slowly, if we calculate those distances again, we reduce the error. What does this actually correspond to? Well, what we've effectively done, or what the machine learning algorithm did for us, is find the values of parameters W1 and W2 here that describes this linear fit best. So initially, those values were very random and our linear fit wasn't that good, but then our machine learning algorithm is trying to reduce this error that we've discussed and arrive slowly at better and better values for W1 and W2. Now, of course, in our scenario, it was a little bit different. We have many genes, not just one variable x. So that's already a higher dimensional setting. And we also have many features or characteristics that can describe this electrophysiological behavior. So it's not going to be just two parameters that can do the job, but I'll leave that as a teaser for the next session where we'll discuss how exactly we can do that.
Thank you very much, Yves. So could you maybe elaborate, like, why would you even use an artificial intelligence to do the calculations? And you explained that the algorithm or the AI tries to reduce the mistake. How is that working? How does the AI do that?
Yeah, again, a good question. Let me then maybe briefly go back. So this, first of all, this is a pretty simple setting that we've had in the beginning. And in fact, if there's mathematicians in the audience, they would actually know that there exists an analytical solution, we call it, for W1 and W2. You can just write down the solution. Here, however, I've created a machine learning algorithm that illustrates step by step what it could do. Because when our problem setting is more difficult, more high dimensional, as we're going to discuss, we cannot just write down the solution like that. So we need a machine learning algorithm to do that job for us, and it can do it pretty fast. So that's why maybe a machine learning algorithm. Yeah.
And if I remember correctly from school, this is like a linear function, but there are other functions like quadratic functions. So why did you choose to use like a linear function to describe this problem?
So again, top-notch question. This linear function is basically a statistician's choice. But nothing would stop us from maybe imagining a quadratic function that indeed could explain this collected data better. In fact, if it were to be a quadratic function, we would have w1x squared plus w2x plus w3. So then there's 3 parameters to tune, 3 parameters that our machine learning algorithm would have to try to find out. And perhaps we would indeed arrive at an even smaller error.
So before you explained that there are a lot of parameters, and you're going to talk about that now. So I assume this is something we're going to hear about in the next part. And this concludes the second round of our questions. Yves, I think your time starts again now.
All right. So in our toy setting, we had a variable x explaining another variable y by tuning parameters w1 and w2. But in our setting, it was more high-dimensional. We have many genes. For instance, 1,000 genes. P-val B is just the name of a gene, could be 1. It is expressed by a certain amount. KCNC2 is another one, also expressed by a certain amount. In fact, if I illustrate it with just 3 genes, it could be numerical values like 1, 0, 2. Now here we had parameters w1 and w2 that were playing with x to arrive at a y. Now what could we do with this? Well, we could multiply it with a matrix. So some of you might painfully so recognize this from high school. But that is essentially how we can transform the gene expression values to other values. And in fact, nothing needs to stop us there, really. We can transform them a little bit more and, in fact, do another matrix multiplication with them, arrive at other values. And I think you're slowly maybe getting the idea. We can do that many times. And as such, we would arrive at final values that we actually hope to be close to features that describe what we electrophysiologically see. For instance, the firing rate, which describes how many of these peaks we see in a certain time interval. I have given random examples of what those matrices could be, but essentially these are now the values that our machine learning algorithm needs to find out. And when our machine learning algorithm has found those out, well, then we have found our mapping, then we have found our function f. Great. Now let's zoom out. So we now again talk about the neurons. We maybe have here, say, 4 different neurons. They look different morphologically. They express genes, perhaps somewhat differently. But we now have a function that can take in those gene expression levels and in those 4 cases predict what it thinks or how it thinks the neuron would respond if the experimenter would come there with its current. In fact, for the blue neuron, it might say, well, based on what I see here, based on how highly certain genes are expressed, I think the peaks will happen, but then maybe there's like a lag phase, happen again, and so forth. Maybe for the pink neuron, the genes, based again on how highly there the genes are expressed, the peaks are somewhat more rapidly in the beginning and then slowing down. Red neuron, maybe it's just 1 peak based on its gene expression levels. For the green neuron, maybe there's a steady firing of peaks that happens. And as such, we figure out, or we're trying to figure out how different neurons are based on gene expression levels, electrophysiology, and morphology, and try to build back the brain.
Thank you very much. You were talking a lot about mathematics, like very, very hard to understand mathematics maybe. Also, there's gene expressions and electrical signals. But how exactly are we going to build a brain out of all of this information?
Yeah, so indeed, I talked about now the electrophysiology, the gene expression levels, the morphology, and those really are important to understand the cells. But indeed, I haven't told anything about how we then would slowly build back or up the brain. So there's indeed other researchers that look at what we call the connectome. So that would be how the different neurons are connected. And then we also try to figure out where exactly those neurons are. And those are indeed essential ingredients to know how then the neurons connect to one another, how they might perform their computations that are necessary. And as such, you can imagine that, okay, we understand the different neurons, so these are building blocks, we know how they are connected, we know where they are. Well, that's a circuit. And maybe from there, bottom-up approach, this is called, we can try to think of circuits. And as such, slowly try to figure out or understand what our brain does.
So you say, like, basically everything is connected. So if you had like a few information on my neurons, could you like using the algorithm, predict how my other neurons would maybe react if you like inserted some current or something like that?
In fact, this is what a machine learning algorithm, as you described it, tries to do. If I go back a little bit, so say I have collected some of your neurons in the brain, and of those neurons, I know the gene expression levels, and I know how they electrically behave. Well, that's collected data. And that collected data I can now use to train or figure out these, or all the values that describe these matrices over here. And so when our machine learning algorithm has then trained this neural network, this deep neural network, well, then our function again is set. And in principle, then I could collect a new neuron from your brain. And I only need to know the gene expression values, because this function, this mapping, will already give me a guess of how that neuron will electrically behave, without even actually having done that experiment.
Okay, wow, that's already a lot you can do with this function. And also, if I remember correctly, in the beginning you were talking about that your research might be able to advance research on like diseases like Parkinson's or Alzheimer's. Well, how is that?
Yeah, so this comes from an understanding that, well, to understand these diseases better, and in fact to maybe understand better what happens correctly in healthy individuals and what maybe happens wrong in diseased individuals with, for instance, Parkinson's or Alzheimer's or any other disease, it probably makes sense to really get a good sense of what the brain does, to understand what our brain does in healthy individuals and perhaps what goes wrong in diseased individuals. So this is really research that hopefully in the future will advance us towards that stage.
I feel like that sounds like a rather hopeful conclusion to this presentation. Yves, thank you so much for your in-depth presentation today.
Thank you as well.
As we've seen, there's still a lot we don't know about our brains and neurons, about our body in general. But maybe further research will really bring us to a point where we can predict what our neurons are going to do, what they can do, what they can't do. And maybe in the future it will be possible to treat diseases like Alzheimer's or Parkinson, or at least we're going to be a step closer to find a disease for that. And yes, maybe at some point we will be able to build an artificial brain. Maybe not to have like a cute AI humanoid friend, but to help researchers like Yves to find solutions for questions they are having. Have you expected that we don't know so much about ourselves? And don't you also think that it's kind of meta that we use our brain to build an artificial brain to find out more about our brain? Let us know your thoughts in the comments below. We're really looking forward to reading them. Finally, once again, a big thank you to Yves for today's presentation, as well as to everybody who was part of the production, to Campus TV, to the Zentrum für Medienkompetenz, especially Michael Seifer, who supported us and oversaw the technical part of the production. And thank you to everybody who was watching today. I really hope you enjoyed today's show as much as I did, and I hope to see you next time at the Science Slam Challenge. Until then, have a good one. Bye.